the exact moment the body decides to start aging. Most people assume aging is something that creeps up gradually. A gray hair here, a slower recovery there. Something that announces itself around 40 or 50. The biology disagrees. Aging begins, in measurable documented cellular terms, at 25. Some researchers now say earlier. What aging actually is. Before the timeline, a definition. Aging isn't the gradual accumulation of damage the way a car rusts. That's a popular metaphor, but it's incomplete. Aging is, at its core, a failure of maintenance systems. The body has extraordinary repair mechanisms, DNA repair enzymes, immune surveillance, protein quality control, telomere maintenance, senescent cell clearance. Aging is what happens when those mechanisms start running below capacity. The damage doesn't increase suddenly. The repairs slow down. And the slowdown starts earlier than almost anyone expects: the cellular evidence. At the cellular level, there are three primary clocks of aging. The first is telomere shortening. Every time a cell divides, the protective caps on the ends of chromosomes, the telomeres, get slightly shorter. They are like the plastic tips on shoelaces. When they are gone, the chromosome frays. The cell stops dividing properly. Telomere shortening begins at birth, but the rate accelerates in the mid-20s as cellular turnover is still high and the protective enzyme telomerase begins to reduce in activity. The second clock is epigenetic. Every cell contains the same DNA. What makes a skin cell different from a brain cell is not the DNA itself, it's which genes are turned on and off. This switching system is called the epigenome. It's regulated by chemical tags on the DNA. Over time, these tags accumulate errors. Genes that should be on get silenced. Genes that should be off get activated. The cell loses its precise identity. David Sinclair at Harvard, one of the leading aging researchers in the world, argues this epigenetic noise is the primary driver of aging, not the DNA damage itself. His lab's research shows the epigenetic pattern of a young mouse can be partially restored in an old mouse, reversing measurable aspects of aging in the process. This research is preliminary, but it has shifted how many biologists think about what aging fundamentally is. The third clock is senescence. When a cell is too damaged to divide safely, it has two options. It can self-destruct, a process called apoptosis, or it can enter a state of permanent dormancy called cellular senescence. Senescent cells stop dividing, but they don't stop being active. They release a cocktail of inflammatory signals called the SASP, the senescence-associated secretory phenotype. In small numbers, these signals serve useful functions: wound healing, tumor suppression. In large numbers, they poison the tissue around them. The accumulation of senescent cells, zombie cells some researchers call them, is one of the most documented causes of age-related tissue dysfunction, and their numbers begin meaningfully rising in the late 20s and early 30s. The 25-year turning point. The convergence of these three mechanisms has a measurable peak and a turning point. Human growth hormone peaks in adolescence. Cellular repair capacity peaks around age 20 to 25. The immune system, specifically the thymus, the organ that produces T cells, begins shrinking and declining in function from the early 20s onward. A study published in Nature Aging in 2021, using proteomics to measure thousands of proteins in blood across age ranges, found significant nonlinear shifts in the protein composition of blood at three key ages: 34, 60, and 78. The shift at 34 was the earliest measurable evidence of systemic aging in the blood, but the researchers noted the underlying cellular changes that produced that blood shift had been building for a decade prior, starting in the mid-20s. The body doesn't announce this. No alarm sounds, no visible change appears. But the maintenance crews start showing up with smaller toolkits, and the work order backlog starts growing. Why this matters after 35. By 35, the accumulated cellular changes have become measurable in everyday function. Recovery takes longer. Not dramatically, but longer. Muscle mass requires more deliberate effort to maintain. Sleep quality begins to shift. Cognitive processing, specifically processing speed and working memory, shows early statistical decline. None of this means 35 is old. That framing is both wrong and unhelpful. What it means is this: the interventions that slow aging, sleep, exercise, caloric moderation, stress management, are not damage repair. They are maintenance system support. They work better the earlier they're applied because they're working with systems that haven't yet degraded severely. The body's aging decision wasn't made at 40 or 50. It was made in the mid-20s, quietly, at the cellular level. Most people spend decades ignoring it. The ones who live longest are the ones who started paying attention before they had to.